these drugs are really, as they seem to be in very limited, small clinical trials, they really are useful in these difficult to treat psychiatric conditions, neurological conditions, possibly even other inflammatory conditions. How can we learn more about their physiology in an ethical way? You know, we're in this constant conversation with society, for better or for worse. And I think things have changed for the better, and I hope that they continue like that. It's always good to be cautiously optimistic.
Hello everyone, Suhaib here. Welcome back to another episode of Citizens Gambit. This episode is a bit of a detour from politics. For this episode, we will be talking about psychedelic drugs. Joining me for this discussion is Dr. Evan Kyzar. Dr. Kyzar has recently completed his MD PhD and is now a psychiatry resident and Leon Levy Fellow in Neuroscience at Columbia University. His research interests include the epigenetics of alcohol addiction, the genetic mechanisms of developmental disorders, and the behavioral effects of hallucinogens. Kyzar has authored numerous publications on psychedelics, including in 2012, Effects of Hallucinogenic Agents, Mescaline and Phenylcyclidine on Zebrafish Behavior and Physiology, and in 2017, Psychedelic Drugs in Biomedicine. In our discussion, we talk about the history and past stigma surrounding psychedelic drugs, the current state of research in the field, the psychological and chemical effects of the drugs, and the future of psychedelic research. So without further ado, I hope you enjoy this discussion with Dr. Evan Kyzar. I'm here with Dr. Evan Kyzar. Finally, we got you. It's been a while, but thank you so much for being here.
Yeah, happy to be here.
You're somebody who I've been wanting to talk to because you're one of the only people who I could get a hand on who has done research related to psychedelics and who has a longstanding interest. And you have multiple publications about the effect of psychedelics, and we'll get into that in detail. And so I was wondering if we could start by talking about your origins, how you got around to being interested in this topic. And so with this episode, we're going to be talking mainly about your publication from 2017. It's called Psychedelic Drugs in Biomedicine. And so to those who are watching, I'll put the link in the description. I'll put the link as well on our blog, and so you can check it out. This is probably the first time you gotten a plug for your for your publication, but
yeah, I mean, for that publication in particular, it's been a popular one. But yeah,
right.
That might be the first time I've gotten like an on-air plug.
Yeah, right. It was great. I was just wondering if you could talk about so your origin and also what prompted your initial interest in psychedelics and just what gave rise to that publication specifically.
Absolutely, yeah. So my name is Dr. Evan Kyzar. I am currently a resident physician in psychiatry and the Leon Levy Fellow in Neuroscience at Columbia University in New York City. I finished my MD PhD degrees at the University of Illinois Chicago. And I guess where this story kind of starts is at Tulane University, where I did my undergrad training in neuroscience. And back at Tulane, we were talking about this earlier. I entered college and wasn't really sure what I wanted to do with my life. I had this interest in medicine that was like purely speculative. I had very little actual hands-on experience in medicine. I remember getting to medical school and be like, thank God I love this, because I had no idea what I was getting into.
I think that's most kids, to be honest.
Yeah, so I'm glad that I had made that decision. But yeah, so I was very interested in the arts and the classics, and I really racked my brain over whether or not to major in something sort of biological and do pre-med, or to major in something like English or the classics or philosophy, something I had just these sort of long-standing interests in. And I took my first Psych 101 that was just to knock out a credit freshman year or something like that. And started talking about the biological bases of behavior, and I loved it. I walked up to the teacher after class, and I was like, I really like this, and sort of explained my dilemma. And she goes, well, if you like this, there's the sort of intro to the neuroscience major class that's taught next semester, and my old mentor taught it. Why don't you take it and see what you think? And just from the first class of my neuroscience coursework, I fell in love. It was this perfect combination of hard science and hardcore physiology and psychology and thinking about philosophy of mind, pulling together, I think, these different strains of thinking and ways about thinking about problems that ranges from the very basic molecular all the way up to constructs that we create in our mind, I think. So I fell in love with neuroscience. I decided that I was going to go to medical school. And I decided that I was going to check off that box that people need on your medical school application and work in a lab. And I applied to a number of different labs, and I saw a publication by Alan Kalueff's lab, who was at Tulane at the time. And it was a recent publication of the acute effects of LSD, lysergic acid diethylamide, in adult zebrafish. And 19-year-old me thought, that sounds like the weirdest and coolest thing that I've ever heard.
Yeah, fish on acid.
Right, and I, through this long-standing interest in philosophy, I had read, various works from people in the beat poet movement, describing the effects of some of these drugs, and I thought it was really interesting. I went and met with Dr. Kalueff, worked in his lab for a summer on a project on MDMA, testing MDMA in zebrafish, just looking basically for sort of acute markers that might indicate anti-anxiety effects, antidepressant effects on things like cortisol, which is the main sort of stress hormone that circulates in the HPA axis of both fish and mammals. Finding some really interesting stuff that generally a lot of these drugs had this sort of anti-anxiety, antidepressant profile. And, we thought that was really interesting. The great thing about doing zebrafish work is it does not take a lot of money. We did have a grant through the National Institute of Drug Abuse, NIDA, through the NIH to do this work. But again, it doesn't take a lot of money to do this. And I think through that entree, through sort of like this behavioral neuroscience standpoint, and reading some of the literature that people like Robin Carhart-Harris at the Imperial College of London and David Nutt, his colleague, were doing sort of simultaneously on a much larger scale in humans, looking at the neuroimaging effects after LSD and psilocybin in healthy volunteers. We just kind of started formulating this idea of how psychedelics work, which is, obviously a huge question, and a question that I think a ton of scientists have spent careers on. And I, unfortunately, don't have the ego to say that I've got anywhere near solving, or that I could get anywhere near solving in my career. But I think what we hope to accomplish in the publication in 2017 in Trends in Pharmacological Sciences, which actually happened when I was out of Dr. Kalueff's lab in my PhD, which was not on psychedelics, which is, we could talk about that in a little bit too. But what we wanted to accomplish is kind of to all this research that has started to pepper the literature on psychedelics. There's really been a revival in interest in these drugs, and in people sort of being willing to conduct research. Research doesn't happen in a vacuum. It happens amongst society, and we're affected by society. I think, the COVID pandemic and us having this over Zoom and things like the drug hydroxychloroquine and the debacle that happened over that, shows us that science is always in a constant conversation with society. And only recently have I think people been bold enough to ask these questions again. And I'm really glad that. So what we tried to accomplish is sort of synthesize that research and pose some questions moving forward for how we can get closer to a heuristic construct of how psychedelics have these long-lasting effects on behavior, with the goal of, if these drugs are really, as they seem to be in very limited, small clinical trials, they really are useful in these difficult to treat psychiatric conditions, neurological conditions, possibly even other inflammatory conditions. How can we learn more about their physiology in an ethical way? I think that's going to be sort of the question for this specific field and niche moving forward.
Yeah. Yeah. I in terms of the interest, initially, I do relate to that sense of integrality that you wanted out of neuroscience, or that you liked, I guess, from neuroscience, the blending philosophy of mind and pharmacology. And that's why psychedelics is so interesting to me, is that you have such a wide range of experiences related to consciousness, related to the self. And we'll get into that. We'll get into that for sure.
No, yeah, and I think that is one of the things that psychedelics really brings to the study of neuroscience. So many people are interested in it either from sort of a theoretical perspective because it alters the cognition and behavior in such a marked way. I mean, it really just changes the way that people perceive the world around them, or they have direct experience with it and they've seen it themselves and they felt it themselves. And most of those people are not neuroscientists themselves, but it causes one obviously to introspect and to think a little bit about what could be driving these things, right?
Yeah. Yeah. And so you touched on it, and we should get into the current state of psychedelics and how the history of psychedelics has led to this point. And you get into this in your in the paper.
Yeah, and I have to give credit again to Dave Nichols, because he wrote large swaths of that sort of history of psychedelics, and you can feel just how personal it is. When I write about the history of psychedelics, I often feel like a voyeur, like I'm getting to do science, do work on psychedelics during this wonderful revival, and people are generally open to these ideas. Dave really lived through it, and I love that. So in we talk about in the article sort of the modern history of society's dalliance with psychedelics starts with Watson's publication about psilocybin-containing mushrooms, specifically in Latin America, in the 40s, and sort of after that, and in the 50s and 60s. It became this thing for sort of literary minds and philosophical, philosophy-oriented minds to write about and to take. It was a method of exploring human consciousness and human cognition in a very personal way. The writings from this period are extremely, you can tell that people were trying to get at how this was actually working, what was going on. Right. And so I think that the 50s and 60s were sort of the first boom in psychedelic research, really when these were first came out. I mean, you can get these drugs anywhere. You go and search the literature. Physicians and psychiatrists and psychologists were using psychedelics in just whatever they could throw it at. They were throwing it. Alcoholism, other types of drug addictions, heart failure, COPD. Just you name it, and someone tried to give LSD to fix it. Like most studies in the 50s and 60s, these were not well controlled, and small trials, things that we can't draw a huge amount of inferences from today. LSD, as most people are probably aware, became the sort of symbol of the counterculture movement in the 60s. And that sort of culminated in sort of the backlash to the counterculture movement culminated with the passage in 1970 of the Controlled Substances Act in the United States, which had been pushed very hard by Nixon's administration and by other administrations after him. And basically the idea was, any drug for any reason was associated with the counterculture movement or could be thought of as harmful to society, was placed under the Schedule 1 category. I think one of the things that people don't understand about drug scheduling most commonly is that drug scheduling has almost nothing to do with science or medicine. There's no hard, medical endpoints for a drug to be considered Schedule 1, Schedule 2. It's all very subjective. Basically, the Schedule 1 drug can have no medical benefit. And obviously that something having medical benefit is a subjective term.
Right.
And so, basically what happened after 1970 with this top-down backlash to psychedelic drugs, to the Controlled Substances Act, and the government's unwillingness essentially to fund research on those drugs. And this was also, you could also apply this to cannabis, to THC.
Yeah, also Schedule 1, right.
Also Schedule 1, also made Schedule 1 with the Controlled Substances Act. And, you could still do research on cocaine and morphine, heroin, basically anything where the expected outcome were going to be that you were going to find that these drugs are bad.
Yeah, right.
And so in the 70s, you see this sort of precipitous drop-off in scientific papers related to psychedelics, pretty much at all. And then in the 80s, you had this very small group of laboratories who were really interested in the effects of these compounds and kind of reading through the papers in the 50s and 60s. They developed this hypothesis that LSD, psilocybin, which is the active molecule in magic mushrooms, mescaline, DMT, that these were all having their effects via the serotonergic system.
Yeah.
In the 80s in pharmacology was the era of receptor cloning, trying to figure out which drugs bound to which receptors.
Mm-hmm.
And so David Nichols, among some of his contemporaries and colleagues, devised this really brilliant, I think, plan of doing research on psychedelics while pretending not to do research on psychedelics, which made it fundable. It's just, it's in hindsight, it's sort of remarkable what they did. And what Dave found, along again with some other laboratories and many wonderful collaborators, is that psychedelic drugs were principally having their main effects through the serotonin 2A receptor, 5-HT2A receptor. Yeah. This was made possible by advances in gene cloning, by specific agonists and antagonists, particularly a drug DOI, which is a psychedelic drug. It is a specific agonist of 5-HT2 receptors. And it, very importantly, is not LSD. Right? So it wasn't necessarily that the funding agencies had an issue with people doing research on psychedelic drugs. They just had an issue with people doing research on LSD. And to a lesser extent, psilocybin. And, I think that really, people just didn't do it. It wasn't a thing that you did. Even when I was doing research in Alan Kaluev's lab, his lab was sort of established at Tulane around the time that I started there in 2008. I joined the lab in 2010. And most of our work on psychedelics was done between 2010 and 2012. Even then, you could sort of count on two hands the number of laboratories that were doing psychedelic drug research. And you could count on basically zero hands the number of people that were funded through the NIH or through another government mechanism to do that. People got at it again in interesting ways. Brian Roth at the University of North Carolina is a structural biologist who fairly recently published the structure of LSD bound to 5-HT2B receptor, which is very similar to 5-HT2A receptor. And the way that he did that was through a very large structural biology grant, not necessarily through an LSD grant. So people essentially had to find ways around getting at these questions. And then I think part of it was Robin Carhart Harris's work and David Nutt's work. David Nutt has been a huge advocate for the field. He published a paper in the Lancet sometime 2000s, basically saying that by any subjective measure, psychedelic drugs and MDMA and cannabis were much, much less harmful to society and to the individual than tobacco, nicotine, alcohol, and so forth. He was fired for his job with the British government for that paper, of course. But he went on to start this group at the Imperial College of London that has been doing research on psychedelics essentially ever since, showing some really amazing effects, culminating most recently in showing that psychedelics have efficacy in treating treatment-resistant major depressive disorder. So now I think you have this coalescence of people are starting to show in small clinical trials that psychedelics may be useful in treating addiction, they may be useful in treating depression, and there's some labs who are starting to get bold enough to do basic research on psychedelics and on MDMA, which is closely related and suffers from the same stigma for the most part.
Right.
When you look at our paper, there's a graph of PubMed results for LSD and psilocybin. And LSD has this huge peak in the 50s and 60s and then drops off
Huge dip.
right as the Controlled Substances Act happens.
Yep.
And psilocybin's like tacking along, not a lot of publications. And then around like 2010, which is right when David Nutt and Robin Carhart Harris started to do their thing, and close to around when Dr. Pulip and I and many other colleagues are working together in certain worlds, you see psilocybin start to tick up. And, you ask what I think is an amazing question. It gets at just the ridiculousness of this whole thing.
Yeah.
Why are people publishing more on psilocybin and not LSD? The answer is people are publishing more on psilocybin because it's not LSD. It doesn't have the same weight. It doesn't have the same stigma. When you walk up to a layperson on the street and ask them what psilocybin is, they likely won't be able to tell you. Maybe more so now, people would be familiar with it. But you ask them what LSD is, and they absolutely know. And they say, "Oh, didn't it make that one guy jump out a window, or doesn't it make you want to kill your dog?" All of this stuff that is really representative of the stigma that we have against these drugs. And I can't reiterate enough. We don't do this in a vacuum. We're in this constant conversation with society for better or for worse. And I think things have changed for the better, and I hope that they continue like that. It's always good to be cautiously optimistic, though, I think.
Right. Yeah. And it's funny because LSD is probably among the more famous psychedelics is not the one that would deserve that treatment, right? I mean, MDMA has been, I guess there's some literature about it possibly being neurotoxic and being a dopamine, for being a dopamine activator, reinforcer, but.
Yeah. So yes, I agree with you. We don't have a lot of great literature on chronic effects of LSD. We do, thanks to, I think, the rave subculture and the real proliferation of MDMA in that subculture. We have a decent amount of data on chronic use of that. And it seems like chronic use, and we're talking about greater than 100 lifetime ecstasy tablets, and I'm not sure what that contributes to a molecule. But in powdered ecstasy, but whatever the equivalent of that would be, does seem to cause atrophy in dopaminergic structures in the brain and cortical structures. The extent to which that is related to MDMA itself and not the contaminants that are typically found in, ecstasy pills or Molly powder is still unclear. There was a really famous study, and I think you might have kind of been getting at this. The Ricard lab at Johns Hopkins, which is a really famous, super
That's
famous.
What I was thinking about. Yep.
Yeah, so a super famous lab that has done work on drugs of abuse, typically looking at negative or adverse effects of cocaine and other drugs that target the dopaminergic system. published a study in Science in 2002 showing that a single dose of MDMA had neurotoxic effects on the dopamine system in rodents. I can't remember if it was rats or mice.
Right. I know there was some controversy about the way that it was carried out. I don't know if you're going to...
The paper was retracted a year later.
Right.
Basically because other labs attempted to replicate the finding and weren't able to do it. And like, weren't just not able to replicate it. Like, they weren't even close to the effects that the Ricard lab were showing. And so, a couple of letters of concern were sent to the editors of Science, and after some consultation with the group that published the paper, they said that they essentially mixed up methamphetamine vial with an MDMA vial. The extent to which that, and the drugs came from the NIH, the extent to which that is possible from NIH provided drugs has been sort of under scrutiny. So the paper was retracted. And in 2003, about a year after it was published, I far be it from me to say any sort of like sinister motive was behind that. Certainly that is like a conspiracy theory type thing that circulates in these circles. I think that there is real concern with repeated chronic use of MDMA. Whether or not you can sort of, pull that out to drugs that act slightly differently, like LSD and psilocybin, is still a matter of debate. It's something that we just don't have enough data on. But yeah, I think that tempers excitement about some of these drugs a little bit, as well it should. I mean, we there's just too much that we don't know. And it comes back to the fact that we just haven't been able to do research on it.
Right. Yeah. This might have a very quick answer, but so far most of the data is that mainly, like, the main receptor is the 5-HTR2A receptor, right? And HT stands for hydroxytryptamine. Are there any other receptors that we know of that are mainly involved? I mean, because that seems to be the main serotonin receptor that's, not even just in your paper, but in most of the other literature as well. Because we know that LSD is a little bit more pervasive in its response, in its response and what receptors it latches onto. But do we know anything else?
The quick answer to that is no, we don't know anything else. But the answer to your actual question is LSD is not a clean pharmacological agent. It has fairly promiscuous binding profile to other serotonin receptors, to dopamine receptors, particularly when David Nichols were doing some of his work in the 80s and the 90s. They found this like very strange sort of what they call like biphasic dose response, where if you trained a rat to distinguish a psychedelic drug from other drugs, and you gave him LSD, but 30 minutes afterwards, right, or 30 minutes before you tested him, pre-treated with a 5-HT2A antagonist, you could block the effects. But then 90 minutes afterwards, if you treated it with a dopamine D2 receptor antagonist, you could also block this sort of like second wave of effects. Is what
wow.
So we call it like a biphasic dose response.
Yes.
It seems to be, so basically the first wave, the first phase seems to be serotonin dependent, and the second phase seems to be at least somewhat dopaminergic, dopamine involved. That doesn't necessarily mean the dopamine receptors themselves are the ones that are being hit by LSD, right? It could be a secondary effect through neurons that have 5-HT2A receptors that projecting onto dopamine neurons, which is something that we've talked about. And David's son, Charles Nichols, who does work at LSU Health Sciences Center in New Orleans, published a really great paper that we talk about in our paper as well, around the same time. I think it was in 2017 or 2016. And they essentially found, using some fairly sophisticated techniques, that there's a very small population of neurons in the brain that respond to psychedelic drugs. It then becomes unclear how this really small population in the brain causes this just widespread, dysregulation or change in behavior and cognition, right? So the thought is, and some of these are located in spots in the brain that are, just exquisitely well connected with other brain regions and with many different brain regions.
Yeah.
And so Charles's group, and then, we adopted it for the review, termed this population trigger neurons, or the trigger population that's activated by LSD, that sort of sets off this molecular and neuronal cascade of events that results in this sort of cortical, just like broadband change in brain activity. And sort of the way that we connected those dots is, LSD binds to the 5-HT2A receptor and has these, and other receptors. I mean, you're right. We just don't know enough. And then goes on and has these effects on a neuronal level, where these trigger populations of neurons activate other brain regions and cause these secondary effects. So like the second dopamine phase that David described back in the 80s. And that basically causes the brain to, form and reform these new connections. The term that Robin Carhart-Harris's group uses is an increase in brain entropy, which, if you're not familiar with the term entropy, just,
Interesting. Wow.
For anyone out there, right, just means basically a movement towards chaos.
Disorder, chaos, right?
Right, yeah, a movement towards greater disorder. And the thinking is, so basically, areas in the brain normally show this sort of predictable pattern of connection with other brain regions. With LSD and psilocybin, those predictable patterns of connectivity between brain regions seem to sort of fall away, and brain regions have patterns of connectivity with other brain regions that they don't normally connect. Is what it seems. And our thought as to why this might explain, and also I think a lot of other people in the field thought as to why this might explain how psychedelics are having this effect on multiple different psychiatric and neurologic disorders, is that often these disorders can be thought of as disorder thinking, where your brain kind of gets stuck in these negative patterns. You can think about it in depression, when someone is continually negativistic in their thinking. Everything that happens gets interpreted in this negative way. There's no way that you can see positives in life. You get stuck. And it's thought, our thought is that maybe these psychedelics are unsticking people's thinking by causing this sort of massive brain-wide reset in plasticity and in connectivity.
Right.
As to, I think this is another casualty of the lack of research in this field. We've had, you've kind of had to pick your battles for a long time. And the battle that people tend to have picked is the 5-HT2A receptor, because the pharmacology is extremely well, it's well known. Thanks to David Nichols and his ingenuity back in a time where it wasn't easy to do this research.
And with LSD, so is this mainly with LSD? I mean, do we see the same effects with psilocybin?
So the great thing about, I guess I should clarify that. With LSD and with psilocybin, they have the same effects on brain connectivity and an increase in sort of brain-wide entropy. They also seem to have really similar effects clinically. So, it doesn't seem like in the few cases where for a given disorder, psychiatric, neurologic, and I think mostly it's depression and addiction that both have been used. It doesn't seem to matter whether it's LSD or psilocybin. Ayahuasca has been used, also for depression, particularly in South America. Ibogaine is a very strange drug that possesses psychedelic-like properties. It's actually been used for opioid detoxification and addiction for a number of years, even decades. There's been clandestine Ibogaine clinics in New York City popping up for probably the past 2 decades.
Yeah.
Um, so yeah, a number of these drugs work clinically, and they seem to work on humans in similar ways. I think the question that we don't know right now is if they cause the same molecular effects. And we know that they bind to the 5-HT2A receptor, and sort of in theory, they should be having similar effects. But each one of these drugs has its own unique pharmacology, its unique off-target effects, and that's a question that's still wide open.
Okay. I think I want to transition a little bit into I think what a lot of people are interested in, which is the visual component, hallucinations. So this has always confused me about psychedelics, about LSD in particular. We know, and you talk about it in the paper as well. Increased blood flow to the visual cortex.
Mm-hmm.
And I guess you also talk about lower amygdala activity, so a lower fear response. Where does so in terms of the content of the hallucinations, I guess where does that come from? How does it tap into? How is that tapped into? How much of it is from events in our life? How much of it is from our past? How much of it is from our own insecurities? And you talk about lower connectivity in the retrosplenial cortex, which leads to like a lower sense of ego, and like more self-awareness as well. And so can you talk a little bit about that? Because I mean, I find it so fascinating, the content of people's hallucinations and how that, in many ways, that's the component that allows them to, I don't know, think of their life differently or take themselves less seriously, and gives rise to a lot of these stories that we hear about psychedelics.
Yeah, we talked about sort of the early days of psychedelic drugs, and the early days of psychedelic research in the 50s and 60s. And while many of these studies sort of don't reach our level of rigor for modern science, they often did a wonderful job of describing the phenomenology and the content of people's experiences on these drugs.
Yeah.
Quite often, the visual hallucinations were much more geometric in shape. You probably can see it. I've got like a drawing of Ernst Haenkel's on the wall back there.
Right.
Very similar to this, like very like geometric shapes. Some often like diamonds, lines, patterns that are extremely basic. You would. I think in terms of, you have to sort of reframe what you think about as a hallucination, right? So the visual content of a lot of people's hallucinations is geometric. And I think part of that is the way that the visual cortex is structured. The visual cortex is structured to identify straight lines, and then lines at various degrees and various angles. Then specific neurons are responsive. The thinking is that really the activity in the visual cortex and the occipital lobe in general is just sort of like a hijacking of global connectivity by these drugs, right? Really, what I think is happening with the way in which this changes people's thinking and like causes them to move forward from the experience with this different perspective. I think it's almost a separate question. And I think right now the best answer that we have for that comes from Roland Griffin's group at Johns Hopkins, who just got this big center. They got a lot of press on psychedelics. Yeah. Basically, Roland Griffin has been doing this type of work, and Matthew Johnson as well, who's there, has been doing this. Yeah, been doing this work for a while, basically showing that the more profound and mystical that your experience is, the more likely it is to lead you to have essentially behavioral change and to look back at this experience as sort of a formative experience in your life.
Right. Positive. Yeah.
Right. And then our question sort of was to, in this review article, was to track back and say, is there anything that we can sort of associate through other people's studies like Robin Carr-Harris and David Nutt's work with these mystical-like experiences? And in Carr-Harris's work, they often use this measure called ego dissolution. So this idea that you feel more one with the world around you, both with objects and with people, with animals, with just everything, feeling less of a delineation between, this is me, I am an entity of will and of volition, and more of a sense of oneness. And that was associated with this greater, connection and activity of the retrosplenial cortex, which has a lot of connections with the posterior cingulate, which we know is involved in basically emotion, response to emotion, and associative response. Posterior cingulate is one of those things that's active when you perceive something new in an environment, maybe a car moving towards you really quickly, and it sort of cycles through, and it's like, we've seen this before, and this is dangerous. So you step back from the road, or you see someone who your big smile on their face, and they're greeting you, and they've got eyes that are quite happy, and your cingulate cortex is one of those things that tells you, this seems like a positive thing, right? It doesn't seem like a threat. This seems like something that could be, a generally positive emotional experience.
Yeah.
And so, really, what I think it taps into is our sense of self in relation to the world in a very basic sense, right? And that's something that philosophers have written about for a really long time. When you truly break it down and think about it, the fact that I look at my hand and I say, this is me, but this is not me, right? There's a component of ego there where it's like, I am this big collection of bones, stacks of fluid, and really gross stuff when you cut me open. But I think that I in some place and have some control over all of this stuff. It's quite remarkable. And I think psychedelics allow us to tap into the neurological correlates of that experience. And when that falls away, when we feel more connected to the world around us,
right.
I think that many people would and I certainly would say that that's generally a positive thing. To have more of a sense of agency, not just over yourself, but over your environment, and also to have a sense of non-agency, and to realize that people around you have that same experience, and this is something that this is an experience that we all share together. Yeah.
The oneness that you described, it's something that that's part of what I was so interested in, is because I've been doing meditation recently. I've started, a few months ago, mindfulness, and to me, that is part of what mindfulness should be, or that is really completely what it should be able to do. You should be able to feel that in that moment, right? That loss of self, and I guess those who can really meditate, right? If you're, if you can do Dzogchen or any other practice, you can attain a similar mental state that we would have if you would take a psychedelic, or if you take something like LSD, or whatever induces that effect in you. And that's a positive thing, right? Because it influences the way that you think about yourself in the world. And so it's viewed as a positive thing. And I said that because I wanted to sort of ask this question, which is more of a philosophical one, about psychedelics. I think you know what I'm going to ask, but so many people benefit from this, and not even just we've been talking about biomedicine and neurodevelopmental disorders, but even those without these disorders, people who take it for just for self therapy and whatnot, they're able to experience aspects of their everyday life. They're able to experience, they're almost hyper aware of the beauty of certain things. And like Huxley writes about this in Doors of Perception, if you've read that book.
One of my favorites, yeah.
This sense of hyper awareness and this loss of self, and it's a beautiful feeling to them, to many. And then when they're out of it, they feel almost like, they feel like they've had that beauty taken away, and some people have a hard time adjusting. Not everybody who does it, but some people do. And that always makes me think, is there something that we're missing about our everyday experience that psychedelics can open our eyes to? Because it's like I mean, like obviously we're stimulating certain aspects of our brain, and they are, as you said with LSD, it's bringing connections together that we normally normally should not be connected. Um, and it opens ourselves up to the beauty of things. And Huxley talks about it in Doors of Perception, in his book. But I mean, is there something objectively positive about the experience that people without these problems can benefit from?
I love that you brought up Doors of Perception and Huxley,
Yeah.
because I'm coming off a 12-hour trip to the hospital, and my brain was not working earlier. I was trying to like recall, I was like trying to pull that back, and I was like, wait, who was that guy? Like, but yeah, no, and I'm so glad that you brought that up, because first of all, Huxley's approach to taking mescaline back in the 50s was so scientific without even meaning to be scientific. And he describes this sense of like you're turning off the reducing valve of the brain that normally like keeps our brain, sort of keeps us blind to all of these different experiences. One of the examples that I always use is, I've been wearing this shirt for a while since I got home from work. My brain no longer processes. I mean, there are sensory connections in my spinal cord running up the spinal tract, saying that I have this shirt on, but eventually the reducing valve of the brain, the thalamus, eventually says, we're not going to worry about this anymore. This has been going on for hours. This isn't important to us.
Yeah.
And Huxley just so beautifully put it, is like you turn the reducing valve off.
Yeah.
And then 62 years later or something like that,
Yeah.
Robin Carhart Harris would find changes in thalamic blood flow after psilocybin.
Crazy.
So it really shows you that like taking this sort of like deep introspective, almost literary approach to these things has like really gave Huxley some insight into what was happening biologically.
Yeah.
I do think that there's a lot of good evidence that psychedelics allow us to experience things in a way in which our brain does not normally allow us to experience.
Right.
As a clinician, I sort of hesitate to put like a positive or negative label on those experiences. I will, as a scientist who can look at and interpret data, say that it's abnormal. It's strange. It's different than the normal. And I think that anyone who's taken LSD or psilocybin or mescaline or DMT at a high enough dose would agree that it's not a normal experience. It doesn't fit within the realm of walking to the grocery store on a Tuesday after work.
Right.
Does it offer us something that we need? Is I think a different question. I think that question is different for every person. I think for a lot of people, psychedelics, and I think one of the reasons people are still very interested in them from a clinical and scientific perspective is the holy grail in science and medicine is a cure. And it's something that we don't have a lot of, a cure, especially in psychiatry and neurology, where we can say you have a disease, here's the treatment. It's not something that you have to take once a day for the rest of your life. It's a cure. It's, that's it. That's the end. Because it causes this just really, really amazing and sometimes lasting behavioral change. It changes your perspective on the world and on the way that you relate to it and on the way that you are as a person. I think that is overwhelmingly a positive thing for the people who it works. I think you hit on something maybe without noticing it or realizing it. A lot of people can sometimes feel this sense of emptiness when the experience is over. And that gets us into something that is important to talk about. And I think it's potential side effects or like not ideal effects of these drugs.
Yeah.
The biggest one is like acid flashbacks, the one that people talk about, hallucinogen persisting perceptual disorder. And I remember when I was in Chicago, I joined a sort of group, and there was a guy, a meetup group just to talk about psychedelics. It wasn't a particularly scientific group. There was a guy who was essentially saying it doesn't exist. That acid flashbacks don't exist and that people are wrong in saying that they're acid flashbacks. And then a paper was published coming off of the Arrowwood database in collaboration with the Johns Hopkins group, showing that something like almost 10% of people with lifetime psychedelics use report some sort of flashback to their experience. And a lot of them report that it was not a positive experience, and it caused them a lot of anxiety.
Right.
And I think we as researchers who have been involved in the psychedelic community and clinicians who are generally open to this as a possible treatment, sometimes criticize people in the past for being closed-minded and saying that, this drug was put on a schedule because people weren't open to the fact that it could have positive effects. You can be closed-minded in the opposite way, where you say these drugs are uniformly positive and uniformly good, and any possible negative effect of it is just fake news.
Right.
I think that's another way that we could get ourselves into trouble. I think that the best thing to do is to listen to people, one, and listen to their lived experience and how they actually felt on, while they had the experience, or before the experience, while the experience is going on, and then after, how long things persisted, and what they felt like their perceptions were. To continue to advocate the government and other funding agencies to allow us to do this research, which I think is really important. I do think that it offers the promise of at least a cure for some subset of people, or something that is better than living a life that essentially offers them nothing. People with treatment refractory depression, really oftentimes are out of options. And so I think that we continue to be able to advocate to do this work, and we're actually able to do this research, then we follow the data. And again, we listen to patients and to people who have been using these drugs recreationally as well, and see what their experiences are, if they're having any sort of negative side effects, if there's anything that we as a medical community, a scientific community, need to watch out for. I think that we talked about the interplay between science and society, and medicine and society.
Right.
Oftentimes, too, that's been a one-way street where medicine has acted in ways that may not have been, seeking the advice of people. We've said in the ivory tower, and we've said we know this better than the people on the street, people who are treating. We need to listen to people, and I think that medicine is becoming more empathetic by the day. I'm really excited to be a part of it, and excited to be a part of what I think is an ongoing revolution in this area. And I think it's got its challenges, but it's a really exciting time to be doing research in psychedelics. Yeah,
wow. Well, I'm excited to be talking to a leader of a supporter of the revolution.
I wouldn't call myself a leader just yet. I don't have enough ranks.
Yeah, right, you're right.
I'm getting.
Sorry, I put you up a few ranks, but it's whatever.
Yeah, thanks for the promotion. I appreciate it.
Right. Yeah, no problem. That was a great way to finish that. And just so we don't go too long, I just wanted to ask, lastly, that being said, with you're saying you're very excited to be doing this in the future. I assume you're going to be going back into this field specifically. I know you have varying interests, range from like epigenetics and other topics. So what plans do you have for your future research? Are you?
Yeah,
I know you're a resident right now, and so you're doing a lot of clinical work.
So I think having the perspective of medicine and psychiatry in particular is going to be actually really important in that, and seeing what things are like, and what people are doing right now on the ground when people walk into a clinic with addiction or depression or schizophrenia and things like that. Getting that training is going to be really important. I think moving forward, I've always had a long-standing interest in just sort of things that drive long-lasting behavioral change on a molecular level in the brain. And so I think psychedelics fit in really nice as a piece of that puzzle, right? They're this drug that LSD is active in humans in microgram doses, which is just
Yeah.
infinitesimally small. You can't really even perceive what a microgram dose looks like.
Right.
And for that tiny amount of something to cause a change in your cognition and your thinking that you refer to as a seminal event in your life for the next 5, 10, 15, 20 years, it's remarkable. And so I do plan at some point in my career on sort of applying these new techniques that we have in science, things like single cell RNA sequencing, circuit tracing and optogenetics and chemogenetics, and turning them on some of these questions. And really the basic question right now that we still don't know is, how does psychedelics cause long-term behavioral change in the brain, or behavioral change through its molecular effects in the brain? We don't know that. I think that'd be a fascinating research question to build, on. And it's something that I have a long-standing interest in, and I hope that when I start my lab, it's one of the questions that I ask.
Yeah. Yes, and I think this episode was really done to help educate people about this topic, and and garner some interest at least in it as well. And so I want to thank you for being a part of this, because it looks like we have to wrap it up just time wise. I will definitely invite you back, because I have so many other questions and things to talk to you about. But again, thank you so much for taking time out of your busy schedule to do this.
Yeah, I hope that next time when you invite me back, I'm not coming off the night shift. But if I am, I will make time. Thank you so much for having me, and I really appreciate it.